In this video, we'll cover the separates-only content in the reproduction chapter. If you haven't already, I recommend that you watch my previous video on the reproduction topic, which will cover the main bulk of the whole sort of chapter. But what we're going to do in this video is, if you're doing separates biology, so you're taking 3 separate GCSEs, then you will need to know a little bit of extra information about the DNA itself. So it's a bit more about the DNA structure and also protein synthesis, so the process of making proteins, and then also the effects of mutations. So first of all, let's look at the structure of DNA. So here you can see we've got a DNA double helix. So I mentioned it in my previous video, but it's a double helix because it's made of 2 strands, and the 2 strands are twisted together into a helical shape, so kind of looking like this. Now, if we look in detail and we pick one part of it, actually we will see that the DNA itself is made up of a smaller monomer called nucleotides. So the monomer nucleotide is made up of 3 parts. We've got the base, which is represented by the rectangle on the right, and then the pentagon in the middle is showing a pentose sugar made up of 5 carbons, and then finally we've got a phosphate group attached to it as well. So nucleotide is made up of these 3 different parts to it. Now, in particular, we say that all nucleotides have the phosphate group and the pentose sugar as normal, but then the base, which is this particular rectangle here, could have slight variations. I could have 4 bases, A, C, G, or T. So it's the sequence of these 4 bases in the DNA that actually codes for the proteins that we have. So the way that this is read is by looking at 3 bases at one time. So when the cell is reading through the DNA and trying to interpret and see what is the amino acid sequence that makes the proteins, they would read it as a triplet code. So what we mean by a triplet code is that every 3 bases is coding for 1 amino acid. So a triplet code would code for 1 amino acid. Throughout the DNA, we've got multiple triplet codes because we've got a long sequence of that. So all of those different sequences together would then make up a protein. So the DNA base sequence is important because we can look at the base sequence to determine my triplet code sequence, and each triplet represents a code for 1 amino acid to make the protein structure. Now we'll come back to how the cell actually does this in a second, but for now let's look a little bit more about the DNA double helix first. So I mentioned that DNA double helix is made of 2 strands and they're twisted together. So essentially, if we look at one strand, this on the left hand side, these two nucleotides are joined together by this bond. And imagine if I've got more nucleotides above and below these 2, then I got a whole chain here. And on the right hand side, I've got these 2 also nucleotides, but you can see that they're upside down. They're going in the opposite direction. Again, imagine there's more of them chained up above and below, but we can then get a separate strand here. And when we draw a DNA double helix, we got these lines in between. That is actually representing some of the bonds that hold the 2 strands together. So we say that there are some weak bonds that can hold the 2 strands like this. Now, in particular, you need to know about the base pairing, because there are very specific rules in how they pair up. Base A always pairs up with base T, or we can have base G, which always pairs up with base C. So this is just an example. So A pairs with T, G pairs with C. Now this rule is what we call complementary base pairing. So the word complementary you would have come across before when looking at the structures of enzymes, looking at the active site of an enzyme. We say the active site has a complementary shape with the substrate, so it means that it's the opposite. This is called the complementary base pairing rule. Okay, so in an exam, they might give you a sequence of DNA bases, so it's completely random, and then they will ask you to write the the sequence of bases in the opposite strand. So, for example, if it's a sequence that's A, C, C, G, T, A, then your answer to it would be the opposite: A to T, C to G, C to G, G to C, T to A to T. So then that would be your answer. It's quite straightforward. So as I mentioned earlier with the triplet code, we know that the DNA base sequence determines the sequence of the amino acids, which then subsequently affects the structure of the protein. So now let's have a look and see how is the cell able to kind of read the DNA essentially and then to produce those proteins. So first of all, you might remember that obviously the DNA is inside the nucleus. So this is what we call the nuclear envelope, and there are actually some holes on the nuclear envelope, which means that stuff can come in and out. Now the DNA itself is too big to leave the nucleus, so what we basically need to do is to make a copy of the DNA that can leave the nucleus to go somewhere else so that it can be read. So in this process, we make this what we call a template molecule. So this template is essentially like a copy of the DNA, which is then small enough to actually leave the nucleus via those nuclear pores to go to the ribosomes, which is where protein synthesis actually occurs. So imagine the template molecule will go from the nucleus and arrives at the ribosome, and it kind of attaches to the ribosome like this. Now the size is not to scale, and it's not the point here, but essentially the key concept that you need to remember is that protein synthesis occurs on the ribosome. So in this template, it's made up of a sequence of, again, bases, which needs to be read by the ribosome so that they can determine what the triplet codes or the codons are to determine which amino acid it should receive. So we've got these other chemicals called carriers that would carry the amino acid to it, and it will pair up with this as well to make sure it's got the right amino acid. So for example, I've got these two chemicals here. So the red pitchfork looking like chemical, that's the carrier molecule, and the orange molecule on top of it, that's the amino acid. So basically there is, like I said, there is a very specific base sequence on the template molecule, and the three bits pointing out from the carrier molecule are also made up of bases. So they can pair up by complementary base pairing to decide which amino acid is the correct one to bind first. So it's kind of essentially like reading the barcode of when you're trying to receive a parcel, then you need to scan the barcode to make sure that the barcode is delivered or received by the correct person. So for example, let's say that this one here is the first correct one. So they were going to move to the template molecule and kind of bind to it and stay there. And let's say if this is the second one, then it will also move and bind to the template molecule like so. And the process basically carries on with more carrier molecules. Now, when the first two, let's say, bind to the template, what happens is that the amino acid would break off from the template from the carrier molecule, and then it will be able to bind itself to the second amino acid like this. Then what happens is that the ribosome would move along the template molecule, and then I would have the third template molecule bringing its amino acid and binding to it again like this. This template molecule will then now leave, and they can then pick up another one. And what happens is that these two amino acids will then break out from the second carrier to bind to the third one. So essentially it's just going to transfer to the third amino acid, and then this process continues. So imagine if I have a few more carrier molecules with other amino acids, they will then again pair up along the template until all of it is transferred. So let's say, for example, that this one is the next one to bind, then again this whole chunk will then break out and then bind to the third one as well. So imagine that this one is now broken off and it will attach itself to the third one. Then this this carrier molecule moves away, and then this process continues onwards like this. So this is going to continue until the whole all of the amino acids are joined together. That becomes the protein. So this summarizes the process of protein synthesis. Just to do a very quick recap, in the nucleus we've got our DNA. First of all, we make a template molecule from the DNA that is small enough to leave the nucleus through the nuclear pore. Then a template molecule would bind itself to the ribosome, which is then allows it to be read, and the carrier molecules would bring the correct amino acids and reading the triplet codes on the template molecule so that they know which is the correct amino acid to bind. Then the amino acids will join up all together to form the actual protein that is being made. So when it comes to exam questions, the key things you need to mention would be the template and the carrier molecule. You might want to mention that it happens on the ribosome as well. The other thing would be to say that every 3 bases code for 1 amino acid. So essentially the carrier and the template are going to be reading by triplet codes to determine which amino acid is the correct one to bind. So that could be like an easy 3 to 4 mark question in an exam. So let's say, for example, that this is an example of a DNA sequence. You can see that if we read every 3 bases, so AGC codes for 1 amino acid, TTA codes for the second one, and CGC, and AGG. So for example, this DNA sequence will then code for a total of 4 amino acids. And there will be many different combinations of amino acids, which is why this sequence is so important. Now the thing is, let's say, for example, that they are all coding for very different, 4 completely different amino acids. So let's say it codes for, this one codes for this amino acid, then it codes for this one, then it codes for this one, then this last triplet codes for this particular amino acid, okay? Now, that would lead to a functional protein. Okay, so for example, ultimately, if I've got loads more together, then I can make a protein that has this particular shape. However, sometimes mutations can happen. So what we mean by mutation, there are many different types of mutation, but mutation means a change in the DNA. So in an exam, you can say it's a change in a DNA base sequence, or it's a change in the gene. It could be any of those things, or change in allele as well. That's also fine. So let's say in the beginning, I still have AGC, TTA, like that. And then suddenly, that when we were supposed to have CGC, it mutated to become CAC. So from CGC, it mutated to CAC, and then last one, let's say it kept the same. Now this time, we know that these 3 triplet codes would still code for the same amino acid. However, this third one would actually code for something that looks a bit different. So it might code for, let's say, another circle amino acid. So this time, I have a different amino acid, meaning that the structure of the protein may change. So for example, rather than having this particular shape, it might have this shape instead. So you can see that the active site shape is now different, for example. So fundamentally, what we mean by that is that the mutation, which is the changes in the genes, can sometimes affect the triplet code sequence, which then affects the structure of the protein, which ultimately affects the function of the protein, especially if it's affecting the active site. So we can have many different outcomes here. So one, it could be that it has a complete change in function, so it just either works in a different way, or it may not work at all. Sometimes it could be that the mutation still led to the same amino acid being coded, so it didn't have an overall change in function, so we call that a silent mutation. And actually, a lot of the time, silent mutations do happen, and that's happening in our body, and we don't realize it. But the idea is, remember that mutation is random. It could happen to any one of those bases, and it very much depends on what base is being changed, if it's going to code for the same or a different amino acid. So it can be sometimes hard to kind of see those effects of mutation, but when it does change the amino acid, it will cause a change in the protein structure, which affects the protein function. Sometimes it's a good thing. It just makes it perhaps makes the protein work even better than before, is more efficient. But sometimes that could be bad. So for example, in the case of cancer, it could mutate the protein, the gene, so that the protein made is particularly good at taking up nutrients. So that is what leads to uncontrollable growth, turning it into let's say a malignant tumor, which is what cancer is. So fundamentally, just keep in mind that what mutation means is a change in the gene or change in DNA base sequence, which can affect the protein structure and can affect the protein function. So there you have it. That's the some of the separates only content in reproduction topic. So again, keep in mind, make sure you know what is the DNA structure like. It's made up of nucleotides, which is made up of phosphate group, a sugar, and a base. And every 3 bases codes for 1 amino acid, which is then happening. It's going to be read by the ribosome by making a template molecule and the carrier molecules with the amino acids. And we know within the DNA double helix, there is going to be complementary base pairing where A pairs with T, G pairs with C. And any changes in the DNA base sequence is called a mutation, which can affect the protein structure, which affects the protein function depending on where the mutation occurs.